Hello students. Now the topic we are going to discuss is hematinics. Hematinics are the substances required in the formation of blood. Most important substances required in the formation of blood are iron, vitamin B12, folic acid and erythropoietin. Now for the purpose of simplicity and easy understanding, this topic is covered in 6 parts, that is 6 videos. First part, that is the first video, deals with introduction to hematinics and the process of erythropoiesis. That is a process of formation of red blood cells. Process of erythropoiesis helps us understand how iron, vitamin B12, folic acid and erythropoietin are essential for the formation of red blood cells and how their deficiency results in anemia. Second video, that is part 2, covers physiology of iron, that is absorption, transport, metabolism and physiological functions and excretion of iron. Third video, that is part 3, covers iron preparations, that is oral and parenteral iron preparations and acute iron poisoning, that is iron overload. We will discuss symptoms and treatment of iron overload. Fourth video deals with physiology of vitamin B12, that is absorption, transport, metabolism and physiological functions of vitamin B12 and oral and parenteral vitamin B12 preparations. Fifth video deals with physiology of folic acid, that is absorption, transport, metabolism and physiological functions of folic acid and its oral and parenteral preparations. Sixth video, that is the last video in the series, covers pharmacology of erythropoietin, a hormone produced by kidneys and its role in the formation of RBC. This is first video, that is hematinics part 1. Introduction to hematinics and erythropoiesis. Now in this first video, we will discuss what are hematinics. We will also study the process of erythropoiesis so as to understand the role of hematinics in the formation of red blood cells. Now hematinics are the substances required in the formation of blood. These are used for the treatment of anemias. Main hematinics are iron, vitamin B12, folic acid and erythropoietin. So these substances are essential for the formation of red blood cells. Each red blood cell contains hemoglobin and this hemoglobin transports oxygen to body cells. Now anemia is a disorder of blood characterized by decrease in the total number of RBC or hemoglobin in the blood. Primary cause of anemia is deficiency of essential factors, namely iron, vitamin B12 and or folic acid, or anemia occurs due to deficiency of erythropoietin. Now this is a very important slide. Now we will discuss, we will try to understand process of erythropoiesis. Now erythro refers to red and poiesis means formation. So erythropoiesis is the process of formation of red blood cells. Now erythropoiesis can be defined as a process of formation, development and maturation of erythrocytes from the stem cells. I hope all of us know that erythrocytes are the red blood cells. Now these red blood cells are produced from the stem cells of bone marrow. Now pluripotent hemopoietic stem cells, these are the primary cells found in the bone marrow. These pluripotent hemopoietic stem cells, they are termed as pluripotent because they have the potential to form all the type of blood cells. These pluripotent hemopoietic stem cells, they can develop into natural killer cells, lymphocytes, erythrocytes, monocytes, granulocytes as well as platelets. And therefore these stem cells are termed as pluripotent hemopoietic stem cells. Now these cells divide to produce two types of cell termed as lymphoid stem cells and common myeloid progenitor. Now lymphoid stem cells further divide and differentiate to produce natural killer cells. These cells are similar to lymphocytes and they produce lymphocytes which we know are of two types, T lymphocytes and B lymphocytes. Now common myeloid progenitor, these stem cells are also termed as colony forming unit GEMM. Now here G stands for granulocytes, E for erythrocytes, M for monocytes, and M for megakaryocytes, that is the platelets. So these common myeloid progenitor, they are termed as multipotential progenitor, or they are termed as colony forming unit GEMM, because they can develop into monocytes, granulocytes, platelets, as well as erythrocytes. Now these common myeloid progenitor, they produce burst forming unit erythroid. Now these burst forming unit erythroid, these are the primary precursors which are required for the formation of erythrocytes. And these are also termed as committed progenitor, because these are committed for the formation of red blood cells or erythrocytes. Now these burst forming unit erythroid series, in the presence of erythropoietin, EPO stands for erythropoietin. In the presence of erythropoietin, a hormone produced by kidney, they produce colony forming unit erythroid. Now these colony forming unit erythroid, these further develop into proerythroblast, and these proerythroblasts, they undergo a number of divisions and differentiations, and finally they develop into mature RBCs termed as erythrocytes. Now for the process of formation of these erythrocytes, hematinics are very important. Now the most important agents are erythropoietin, iron, vitamin B12 and folic acid. All these substances are required for the differentiation and division of proerythroblast and their final maturation into mature erythrocytes or RBCs. Now let's study in detail stages of erythropoiesis. That is how proerythroblast finally develops into a mature RBC, that is the erythrocyte. Now total 7 days are required for the transformation of proerythroblast to a mature erythrocyte. Now 5 days are required by the proerythroblast to develop into reticulocyte, and during the process there is a rapid division and differentiation of RBC precursors. Now for this division and differentiation of RBC precursors, synthesis of DNA is very essential, and folic acid and vitamin B12 are very essential for the synthesis of DNA. Therefore, we can say that vitamin B12 and folic acid are very essential for the division and differentiation of RBC's precursors, so that the proerythroblast develops into a reticulocyte. And the duration, the time required for the development of proerythroblast to reticulocyte is 5 days. Now further, once the reticulocyte is produced, this reticulocyte matures into a mature RBC or a mature erythrocyte. Now the process is termed as terminal maturation, and for this terminal maturation also vitamin B12 and folic acid are required. So reticulocyte matures in 2 days to a mature RBC or a mature erythrocyte, and for this maturation again vitamin B12 and folic acid are very essential. Now apart from this, iron is required for the synthesis of hemoglobin. Synthesis of hemoglobin begins in the first stage itself, that is in the proerythroblast stage, and for this synthesis of hemoglobin, iron is essential. Now apart from this, erythropoietin, the hormone which is produced by the kidneys, triggers the process. It stimulates the process of erythropoiesis, and this erythropoietin is responsible for the utilization of vitamin B12, folic acid and iron by the RBC's precursors and their final development into a mature erythrocyte. Now let's talk about these individual stages one by one. The first stage is the proerythroblast stage. So when stimulated by erythropoietin, colony forming unit erythroid, they get transformed into proerythroblast. Now these proerythroblasts, as it is very clear in this diagram, they are very large cells. They are the largest of all the cells. So these are very large cells. Diameter is about 20 micrometer, and as again clear from the diagram, the nucleus is very big, and therefore there is a high nucleus to cytoplasm ratio. And this proerythroblast, it possesses prominent nucleoli also. Now, as it is shown in the diagram, this nucleus, it possesses fine granular nuclear chromatin, and there is a synthesis of DNA. Synthesis of DNA and formation of hemoglobin begins during this stage, that is the proerythroblast stage. Now, in the presence of erythropoietin, vitamin B12, folic acid and iron, this proerythroblast, it differentiates and it develops into a basophil. Now, basophilic erythroblast is produced on day 2. Basophilic refers to basophilic cytoplasm. Now, as shown in the diagram, the color of cytoplasm shown here is in blue color. So, blue color represents basophilic cytoplasm due to huge synthesis of RNA in this stage. And the cell is termed as erythroblast. Blast refers to an immature cell. So, this is the basophilic erythroblast. Now, as it is clear from the diagram, there is reduction in the size of cell. There is reduction in the size of nucleus, and there is complete disappearance of nucleoli. Now, chromatin in this nucleus, it becomes progressively clumped. There is further condensation of nucleus, and there is reduction in the size of cell, there is reduction in the size of the nucleus, and the basophil finally develops into the polychromatic erythroblast. So, this polychromatic erythroblast, it is produced on the day 3. Now, polychromatic refers to the cytoplasm. Cytoplasm is basophilic as well as acidophilic. Basophilic due to the synthesis of RNA, and acidophilic due to increased presence of hemoglobin, and hence erythroblast is termed as polychromatic erythroblast. And further, as it is clear from the diagram, there is reduction in the size of cell, there is condensation of nucleus, and the polychromatic erythroblast, it develops into the orthochromatic erythroblast. Now, in the orthochromatic erythroblast, there is acidophilic cytoplasm due to abundant synthesis of hemoglobin, which is shown here in the reddish color. And during the orthochromatic erythroblast stage, there is final condensation of the nucleus. And once the nucleus is condensed, it is extruded out of the cell, it is pushed out of the cell. And all these stages are occurring in the bone marrow. So, orthochromatic erythroblast, after the extrusion of the nucleus, it develops into the reticulocyte. Now, this reticulocyte, it is produced on the 5th day. Now, it has no nucleus. The nucleus which was pushed out of the cell is engulfed by the macrophages. And on the 5th day, reticulocyte, that is the immature RBC, is produced in the bone marrow and then released in the blood circulation. So, it is released in the blood circulation. It travels to the spleen for maturation, and maturation requires 2 days. And maturation also requires supplementation of vitamin B12 and folic acid. There is breakdown, and there is expulsion of endoplasmic reticulum, Golgi body, lysosomes, mitochondria, ribosome, and finally the reticulocyte, it develops into a mature erythrocyte. So, the process of erythropoiesis is completed in 7 days. 5 days are required for the division and differentiation of erythrocyte precursors, whereas the last 2 days, that is 5 to 7 days, they are required for the terminal maturation of reticulocyte into the erythrocyte. Now, let's here revise the changes which have taken place during the erythropoiesis, as we have already discussed. There is reduction in the size of cell. See, as the proerythroblast develops and differentiate, there is reduction in the size of the cell. The diameter of a proerythroblast is about 20 micrometers, whereas the diameter of erythrocyte is 7.2 micrometers. So there is progressive reduction in the size of cell. There is progressive reduction in the size of the nucleus, there is condensation of nucleus, and finally extrusion of the nucleus. Now, second feature, there is change in the staining of cytoplasm from basophilic to acidophilic, as we have discussed. The proerythroblast, it has a basophilic cytoplasm, basophilic erythroblast, again basophilic cytoplasm. Polychromatic cytoplasm is basophilic as well as acidophilic, and orthochromatic, it possesses the acidophilic cytoplasm due to excess presence of hemoglobin in the cell. And there is a disappearance of nucleoli. The nucleoli are found to be present in the proerythroblast, but when it transforms into the basophilic erythroblast, there is complete loss of the nucleoli. Synthesis of hemoglobin starts from proerythroblast, and a large amount of hemoglobin is found to be present in the cell during the orthochromatic erythroblast stage. Progressive condensation of nucleus, then there is extrusion of the nucleus from orthochromatic erythroblast, and then the stage of reticulocyte. During this stage, during the maturation of reticulocyte, there is breakdown and there is expulsion of endoplasmic reticulum, Golgi body, lysosomes, mitochondria, ribosomes, so that more space is available for the hemoglobin. And finally, the reticulocyte transforms into the erythrocyte. So these are the stages of erythropoiesis. This is how the proerythroblast develops into a mature erythrocyte or a RBC. Now, hope by now you must have understood the importance of hematinics in RBC formation. Now, let's once again memorize role of erythropoietin, vitamin B12, folic acid and iron in erythropoiesis. Now, talking first about erythropoietin, a hormone produced by the kidneys. Now, erythropoietin, as we have already discussed, it triggers and it stimulates erythropoiesis by stimulating colony forming unit erythroid. Erythropoietin regulates differentiation, proliferation and maturation of RBC's precursor. That means erythropoietin is a hormone that stimulates or triggers the process of erythropoiesis. Erythropoietin is responsible for the RBC's precursor to utilize vitamin B12, folic acid and iron for the development of proerythroblasts to mature erythrocytes or RBCs. Now, coming to the role of vitamin B12 and folic acid. Now, as we have already discussed, vitamin B12 and folic acid, their primary role is the synthesis of DNA, and synthesis of DNA is very essential for the division and differentiation of the cells. And further, vitamin B12 and folic acid are also required for the maturation of reticulocyte to erythrocyte. So, vitamin B12 and folic acid are required for the synthesis of purines and pyrimidines, which are the DNA precursors. That means vitamin B12 and folic acid are required for the synthesis of DNA. They are essential for the condensation of nucleus, essential for the division and proliferation of RBC's precursor during differentiation. They are very essential for the maturation of RBC, that is the transformation of reticulocyte to erythrocyte in the peripheral blood. And apart from this, vitamin B12 is very essential for the lipid synthesis. Lipid synthesis for the myelin sheath, that is myelin sheath of the nerve cell. So, for the healthy nerve cell, vitamin B12 is very essential. And iron is also very important hematinic. Iron is required for the synthesis of hemoglobin. So, this is all about introduction to hematinics and the requirement in the process of erythropoiesis. And if you find this session helpful, kindly like, subscribe and share this video. You can ask your doubts in the comment section. I will surely answer all your doubts or all your questions. Thanks for watching this video.